Welcome back for part 3 of my discussion with Dr. Sumit Chanda, Director of Inflammatory and Infectious Diseases at Sanford-Burnham. My name is Dr. Daniel Haters, and I am the Executive Chairman at Model Medicines. Sumit, for part 3 of our conversation, I'd like to discuss our partnership, how it originated, and our joint approach to discovering broad-spectrum antiviral therapeutics to solve both this pandemic and the next one. If you don't mind, I'm happy to describe how the collaboration was started from the Model Medicines perspective. As COVID-19 hit, we at Model Medicines became aware of the inspired multinational, multi-institutional group that you put together, and we followed the team's progress with quite a bit of interest. And then, due to a bit of serendipity, I believe you became aware of Model Medicines and our work with a collaborator in common, Dr. Arnab Chatterjee at Caliber at Scripps Research, who we were working with to validate broad-spectrum antivirals we had discovered for a set of well-known, validated COVID-19 targets. Where the story gets interesting for Model Medicines is that we use a knowledge of fundamental chemistry and machine learning to identify drugs for validated targets. However, we do not currently discover or validate targets. On the other hand, we became aware that your work, post the publication of the Nature paper, became focused on identifying novel broad-spectrum targets. This was a match made in heaven from our perspective. Can you speak about how and why your laboratory pivoted after the publication of the Nature article to focus on the identification of new targets that no one else in the world had identified, and what the potential impact could be from discovery of these targets?
Yeah, I mean, so, one of the things that, we had an advantage for in this pandemic is really the first global pandemic where we can empty out the omics toolboxes to understand the virus, okay? Whether it be high throughput small molecule screening, like the work that we've done, genome-wide CRISPR screening, RNA-seq, proteomics. We learned a lot about not only the virus, but how the virus interacts with the host, okay? However, one of the limitations of the omics field in general, and particularly for the host pathogen interactions between SARS-CoV-2 and the human host, is that there's a lack of synthesis of that knowledge, right? So you get a list of genes, and then you hope that one of those genes has already a known drug target, and then you test that drug target, okay? So there's that kind of one step away from harnessing the omics data, but really and as you know, Daniel, to harness omics data, right? These large data sets. It's not a one-to-one correlation, right? You really need to do sophisticated network-based analysis, and more and more, this is going to be based in AI, right? These are going to be using kind of dimensionalities of thought that a human brain really can't process, that we can't pick out, the patterns within the noise. And I think that's really where the strength of our collaboration comes in that we harness the omics data, which is kind of ground level data, and synthesize it in a way where we can action things that are not empirically obvious, right? So the analogy I use is that even with the omics technologies, we're still looking for our keys underneath the lamppost. And what we really want to do is look for our keys wherever they may be hidden, right? Or wherever we may have lost them. So this is really where I think our partnership here has the ability to expand our not only our knowledge into SARS-CoV-2 host interactions, but illuminate druggable targets that are really not obvious in a first pass omics analysis.
I share your point of view. I am fascinated by the potential what can happen at the intersection of cutting-edge, bench-created chemical and biological information or data sets and our cheminformatics-driven machine learning drug discovery algorithms. When you merge those two concepts together, I believe you have the potential to accomplish something truly unique and truly special for the global population, namely time, quality of life, and hope that no matter what pandemic may emerge or what diagnosis I might receive, there is a significant possibility that science can create a solution. Specifically at Model Medicines, We think about our technology not as a way to replace bench-level science, but rather as a way to make its data actionable today, rather than 10 or 20 years from now, and at a fraction of the cost. In our case, this means standing on the shoulders of the great labs of the world, like yours, to translate your discoveries into new therapeutics on the shelves of your local pharmacy today, not 2 decades from today, a concept that was unfathomable just a few years ago.
Absolutely, I couldn't agree more. It's all about the synthesis, right? I mean, I think that there are there's a lot of power in doing omics, but there are a lot of limitations. And so, working with a team like yours and a technology that you have enables us to really extract meaningful, actionable intelligence beyond, I think, kind of what the obvious things are. Looking at these data sets, right? I mean, you're looking at really extracting emergent properties of the data that can't be deciphered by looking at lists of genes. And so, in our first pass, as you mentioned, the work that we've done, right, we used essentially a fundamental triaging type strategy, right, where we screened 12,000 known drugs and then drilled down to 21 and then tested one in an animal model. And that was really solely just based on a set of criteria that is driven by activity, right? We all know that is not the end all be all, right? I mean, there is the holistic analysis of that large data set. There's more in there than a list of 20 drugs, or in a CRISPR screen, your usually your 10 best things are probably not the lesson that you want to learn from there, right? There are lessons in there that you can't learn by just ranking your data, which is how most people right now action omics data. They take their gene list and say, well, here's my ranking criteria. These are the 10, 20, or 30 best things. But what needs to happen is really if you look at it holistically and then take out things that multiple points in your data all point to, then I think that's where you're going to get the most value. And that's not something I think, human brains are really trained well to do. And so this is where I see the power of AI and then converting that into immediately actionable hypotheses. So I think this is key, right? A lot of people are doing AI and there's no kind of way to pressure test how good those predictions are, right? So the way to do this, in my opinion, is to make these predictions, go back to the lab, test them, feed them back, reiterate and improve your model to a point where you get to a highly predictive set of tools that then you can really open up brand new avenues of investigation that I think no one could really anticipate, even with just analysis of a single data set.
I appreciate that point of view, and I want to focus on something you just mentioned. High throughput screening, from one perspective, is a highly sophisticated, efficient, technology-driven technique to discover new therapeutics that stand as a shining light of human ingenuity. From another perspective, it's a brute force, inefficient technology devoid of hypothesis-driven concepts, and one which typically ignores the entire compendium of human knowledge in chemistry and biology in favor of trial and error horsepower. You were mentioning that your lab screened over 11,000 or 12,000 compounds in your study. On the other hand, hypothesis-driven drug discovery work based on fundamental understanding of chemistry and biology is bespoke, agrarian, and inefficient. At Model Medicines, we had the thought, why does drug development have to be either or? Why can't we use a fundamental understanding of chemistry and biology combined with machine learning to create a new platform that is highly sophisticated, efficient, and technology-driven to discover new drugs? When we use data and machine learning, we can parse and really begin to understand the data humans have already created in a way that was not possible before. For instance, sometimes in a matter of minutes, our algorithms can look at a data set and begin to see patterns, i.e., drugs with this side group or this solubility can, when combined with this side group and this electrostatic profile, preferentially bind a certain target. Don't get me wrong, this type of knowledge and understanding can be encapsulated in a single person who's worked 50 or 60 years in the field. And we typically would say that those epiphanies occur because of a scientist went with their gut or on a hunch, when really the human mind was simulating the machine learning algorithms I mentioned before. But that knowledge is transient and passes into and out of the world with us, oftentimes without being codified or made repeatable. What we are doing at Model Medicines is harnessing all of this fundamental knowledge of biology and chemistry that the world has and making it actionable, repeatable, and efficient. However, to be clear, AI and ML is nothing without that fundamental knowledge and data. At the next level, our mission is to build a technology platform that is not simply, quote, one more tool in the toolbox of drug discovery, but rather something that can truly change the business of the pharmaceutical industry by fundamentally altering the time to market and the cost to market of new therapeutics. We aspire to change 20 years and $2 billion to 2 years and a few $1 million. But again, it is impossible for us to do that without partnering with scientific leaders like you and your lab, who are producing the fundamental data that our algorithms are fed. It is truly a symbiotic relationship.
Yeah, no, I absolutely agree. You know, my experience with modeling and AI is that, you know, if you can provide the rules of the road to the AI, it does much better than kind of an open playing field, right? So, you know, things like driving and chess, where they have kind of finite boundaries, AI is really good at. And so this is what our data, I think, allows, is that here's kind of a roadmap, and let's look at it in a way that is not kind of a one-to-one mapping, and pull out things that are immediately actionable. And you guys will tell us that, and we can go back and predict it and really, you know, validate how good we are. And I also agree that, look, you know, I mean, there's a place in science to be incremental, but that's not what we do, right? We love swinging for the fences. I'm kind of a go big or go home kind of scientist, and so this is why I think this opportunity is particularly exciting.
Go big or go home. I love it. We will let those be the final words of part 3 of our discussion. In the final segment of our conversation, part 4, I want to understand your thoughts about what the future of AI and drug discovery looks like from where you sit. Thanks for your time today, Sumit.